So I'd now like to call up to the podium Kent Hunter, who's going to talk about breast cancer.
So this is going to feel like deja vu. It's pretty much the same talk, but a different tumor type. So my laboratory is interested in breast cancer, and this is the reason why. Some of you have seen this slide before. Breast cancer is the most common malignancy in women and is the second leading cause of cancer-related death. And the important thing to know is that for breast cancer and for most solid cancers, it's not the primary tumor that is the source of the greatest mortality. It's the metastatic disease. And although we are getting much better at identifying those women who are at risk of developing metastatic disease, which women and why they're actually developing metastatic disease is still unclear. So 20 years ago this year, I kind of hate to say that, we set out to try to identify whether or not there was an inherited component involved in all of this process. And I'm going to tell you the latest results from our laboratory for one of the genes that we've discovered. So this is the model we use. It's a transgenic mouse model. It's one of the most aggressive metastatic models available in the mice. MMTV polyomma T antigen. These animals develop mammary tumors with 100% penetrance by 9 weeks of age, by 100 days of age when they have to be euthanized. More than 85% of these animals will have developed pulmonary metastasis. And this is actually a pretty good model for human breast cancer because the polyomma T antigen activates the PI3 kinase pathway, which is the most commonly mutated pathway in human breast cancer. So 20 years ago, we set out to look for inherited modifiers of this phenotype by setting up a strain survey, which I would not recommend people do anything on this scale these days. We took the polyomma T animal and bred it to all of these different inbred strains with the hope of trying to find one strain that actually modified the metastatic trait. And what we ended up with was the following result. Basically, there are lots of modifiers of metastasis available in the mouse genome. And as you can see here, we have strains that are as metastatic as the original strain, which is the FVB background, which the transgene is on, and there are a number of strains down here which significantly suppress metastasis, and in some cases also change tumor latency and tumor growth, but there are individual strains that you can pick out. The only difference in phenotype is metastasis. For the purposes of this talk, we're going to concentrate on this animal, the molossinous animal, MOLF. This is a wild-derived strain from Asia. Introduction of the MOLF genome by breeding into the polyomma T animal suppresses metastasis significantly. It also does suppress growth and latency of these tumors, but we are able to actually separate those out, and I will mention them later, what we're interested in. Like Kendra, we have set up the standard sort of genetic mapping analysis, except we did a backcross rather than the intercross that Kendra was talking about, and we identified a single peak on the end of chromosome 6 that was associated with metastasis suppression. Based on the observation that most polymorphisms is not coding, we decided to look for metastasis modifiers that were driven by gene expression changes rather than coding mutations. So in order to look for candidate genes, of these 194 animals, we did microarray analysis on 134 and looked at the genes within this interval to find transcripts whose expression was significantly associated with metastasis and therefore might be candidate genes. When we did that, we found 12 genes in that candidate interval that were associated with metastasis. Here is the hazard ratio in the molossinous cross and the p-value, and you can see they're quite significant for the most part. To make sure we're actually looking at something that's human relevant, what we have done is taken this gene signature and the hazard ratios and used these 12 genes in a weighted gene signature analysis of human breast cancer gene expression data set to make sure that these things are actually relevant in human, because I'm not really interested in solving mouse metastasis. And when we did that, we found something that was a little bit of a surprise in that, yes, you can use this weighted gene signature, insisting that the genes have the same transcriptional direction and strength in human data sets. And we found that it was predictive in ER negative receptor breast cancer, which is a little bit of a surprise because the polyomma T animal is supposed to be a model of estrogen receptor positive breast cancer. But as it turns out in other work that we've published elsewhere, changing the genetic background of the tumor actually changes the gene expression profiles that predict whether or not it's a model of ER negative or ER positive breast cancer. So it's not just the the somatic mutations that determine whether you are an ER positive or ER negative breast cancer, but it's also your inheritance as well, at least at the transcriptional stage. So the next thing that we've done is started identifying genes and looking at these genes in our various assays. And again, for this talk, we're going to focus on this gene here, Arntl2, which is a circadian rhythm gene right at the top of this peak. But I don't want you to think that this is the only gene here that has an effect. We know that there's at least one other, and probably most, if not all of these genes are contributing to this large peak we see here. But we can only do these really, up until now, one gene at a time. So how have we tested that? Well, it's very easy. In metastasis, you can do this using a lot of cell lines. There are a number of different mammary tumor cell lines that are available. You can take your gene of interest. In this case, I'm showing you an RNAi knockdown vector, but you you modify your gene expression in mammary tumor cell lines that we have. Those cell lines are then transplanted into the orthotopic site, a mammary fat pad in the mouse. You let the tumors grow, and then you see whether the manipulation you've introduced will change the metastatic capacity. When we did this with Arntl2, what we found is just showing you our knockdowns here. We achieved pretty good knockdowns. When you knock down Arntl2 using separate shRNAs, you can see that the metastatic capacity of these tumors dropped. I'm not showing you this here, but in this case, the tumors were not affected in the growth, either the latency or growth rate of the tumors. And you see the opposite effect if you then overexpressed Arntl2, so you're increasing the metastatic capacity, suggesting that just manipulation of relative levels of the circadian rhythm gene is having an effect. The nice thing about doing genetics is you can actually do the experiment in humans. We've then looked for SNPs that are associated with Arntl2 expression in human populations, then examined whether that SNP predicted outcome in human breast cancer patients. And, oops, I'm out of order. I'm sorry. I'll come back to that. We have done human epidemiology. I'll come back to that. But again, going back to look at what's actually affecting, what's the driver of polymorphisms here in the genome. We've gone back to look at the sequence analysis of FVB versus MOLF. This analysis is one of the reasons why Anthony was talking about the MOLF genome the other day. These are the strains that we're interested in did sequencing. Again, as Thomas mentioned earlier, the annotation of these genes are not always particularly good. Arntl2, if you look at the ref-seq transcripts, ends here. That actually turns out not to be the transcriptional start site of that particular protein, because that transcript right here, if you look at the ref sequence, is actually starting at the ATG. If you look at Ensemble, there's another EST that spans this particular region, and there's actually turns out to be a DNase hypersensitivity site over here that would indicate that this is probably the promoter, not over here. When you look at polymorphisms present in this part of the genome, interestingly enough, there are a whole bunch of SNPs between MOLF and FVB that fall right into this DNase hypersensitivity site, which suggests that maybe we're looking at a cis effect due to promoter polymorphisms. We have tested that directly by, as Tyler suggested, doing CRISPR analysis. In this case, we took an FVB cell line that is highly metastatic. We replaced this 900 base pair section right across this DNase hypersensitivity site with the MOLF allele, as you can see here. That results in reduction of Arntl2, so introduction of these 10 to 12 polymorphisms is in fact suppressing the ability of this transcript, this promoter to develop the transcript. And then when you implant that cell line, just like we see with the shRNA, the ability of these tumors to metastasize is significantly suppressed. So we are simply looking at least a contribution of these promoter variants in this particular gene. Okay, now to the human epidemiology. Going back and looking at the SNPs in the in the human genome, this particular SNP RS4964008 was associated with a suppression of Arntl2 transcript in the human data sets in breast cancer by GTEXT. When you look at this SNP and association with ER-negative survival, breast cancer survival in the Shanghai breast cancer cohort, what you see is it is significant. It's modest, but it is significant, and we're seeing exactly the same protective effect that we expect based on this SNP reducing Arntl2 expression, suggesting that Arntl2 is acting as a metastasis modifier both in mouse and human. Finally, people were asking us, okay, what transcription factors is all of this, these polymorphisms changing? So we've done the analysis looking at this 900 base pair region by doing biotin pull downs of that region, looking what these polymorphisms either the different proteins that bind to these two different polymorphic alleles, either presence or absence or reduction by mass spectrometry. And interestingly enough, what we found is that actually no difference in transcription factors that bind to these. That was a little bit of a surprise. What it is different is that there are two chromatin binding proteins, PARP1 and CTCF, that are affected. Here showing the MOLF allele versus the FVB allele. What you're seeing is there's it's a modest reduction, but there is a reduction of both PARP and CTCF binding to these two different alleles. Why is this particularly interesting? Well, I mentioned to you that Arntl2 is a circadian rhythm gene, and serendipitously, while we were puzzling about all of this, a paper was published showing that PARP1 and CTCF binding together on DNA is involved in the oscillation of circadian rhythm transcript promoters in and out of the nuclear lamina, which suppresses transcription. And so what we believe is happening is these polymorphisms are changing the efficiency with which this particular promoter is being moved in and out to the lamina-associated domains in the nuclear membrane. And we know that, although I'm not showing you the data, not only is Arntl2 affected, but the flanking genes are also affected by introduction of the MOLF allele at this particular site. So that's the model that we're currently working with. We're now doing some interesting things, I hope they'll be interesting, about circadian rhythms. And stay tuned, hopefully we'll have some interesting stuff for you in the future. So with that, in summary, we believe Arntl2 is a metastasis promoting gene. This is strongly supported by a paper published by one of Tyler's ex-postdocs, Monty Winslow, who just recently showed somatic variation in Arntl2 affects lung cancer metastatic disease. We see polymorphisms in the human Arntl2 that is associated with progression in ER-negative breast cancer, as we would have predicted. And it certainly seems to be acting through the PARP1 CTCF interaction that may be affecting circadian rhythms and the effect of circadian rhythms on tumor progression. But this is something that we're going to be spending a lot of time looking at in the future. And with that, I'd just like to thank the members of my laboratory and the collaborators at Vanderbilt who helped us with the epidemiology, and I'll be happy to answer any questions.
Thanks, Kent, for a great talk.
So just to follow up on a point you made towards the very end, when you make the substitution into the Arntl2, does that affect the eQTLs of the other genes in the region that you've allowed you to focus on those genes in the first place?
So we actually haven't looked beyond doing qPCR on the flanking genes. That's a good question. We don't know how far the effect will propagate down the chromosome, but that's an interesting thing to look at.
Okay, if there's no other questions, please thank Kent again.